We've learned about cell division, but what about the division of DNA? DNA replication is a multi-step process in which a single DNA molecule becomes two DNA molecules. But how does it work? What is DNA made up of? Well, DNA stands for deoxyribonucleic acid, and it consists of three different components. The first component, the phosphate group. The second component, the 5-carbon sugar. In this case, the 5-carbon sugar is called deoxyribose. The last component, the nitrogenous base. These nitrogenous bases are called adenine, thymine, guanine, and cytosine. Without further delay, let's meet the enzymes that are responsible for the process.
There are 5 enzymes that allow DNA replication to occur. The first one is helicase. Helicase's primary function is to unzip the hydrogen bonds between the paired nitrogenous bases. Next up, we have the single-stranded binding protein. Its function is to bind the regions of DNA and prevent the single strands from closing back up. Next, primase. Primase begins the RNA sequence complementary to the single strands. Penultimately, we have DNA polymerase. DNA polymerase creates the daughter DNA molecules, allowing the daughter strands to be constructed. Last but not least, ligase. Ligase closes the broken covalent bonds between the sugar phosphates and searches the DNA strands for any mishaps. With all the main parts out of the way, let's go to the animation. This is the original DNA strand. Replication begins in an area called the origin of replication, which is represented by the location of where the first bond is unzipped. The first step in DNA replication is the unzipping of the double-stranded DNA by the helicase enzyme. The shape of unzipped bonds is known as the replication fork. The strands are anti-parallel, meaning they move in opposite directions. As helicase breaks the bonds, the single-stranded binding protein prevents the strands from closing back up. Helicase and the single-stranded binding protein are working simultaneously, and even while those two enzymes are working, a third enzyme known as telomerase keeps the DNA from cutting too short. Once the helicase has broken about half of the hydrogen bonds, primase begins to add RNA primers to the leading strand. There is only one RNA primer on the leading strand, and it runs from 3' to 5'. After primase has added an RNA primer, the first type of DNA polymerase adds daughter strands following the RNA primers. As this is happening, an RNA primer is added to the lagging strand, but in different positions than the leading strand. This primer runs from 5' to 3'. This is necessary so the strands continue to be anti-parallel. Daughter strands are continuously added and are complementary to the original DNA strand. This model is known as the semi-conservative and was coined by Watson and Crick. Following this, another RNA primer is added to the lagging strand, creating a sequence known as the Okazaki fragment. This continues until daughter strands have filled up all the positions where they can be added. Then, the second type of DNA polymerase goes through the strand and replaces all RNA primers with daughter strands, leaving the bonds unconnected. After this, ligase goes through the original and replicated DNA strands and creates covalent bonds between the sugar phosphates. Finally, the DNA strands coil and then can be transferred to another cell through cell division.
But what if DNA replication goes wrong? Just like cell division, DNA replication can go wrong. For cell division, when a cell begins to divide abnormally, it transforms into a growth known as what we call cancer. DNA replication is similar, but the DNA becomes mutated if the process occurs too quickly. DNA replication is a very accurate process, but there's one major enzyme that can make the process go haywire: DNA polymerase. If DNA polymerase inserts a wrong base, consequences will occur. One of those potential consequences is the development of cancer. However, this mistake can be corrected before the process is completed. As you already know, ligase is responsible for making sure that all the covalent bonds are connected and all the different bases are in the correct area. Similar to DNA polymerase, ligase can also malfunction, which would lead to this cancer. But on average, DNA polymerase makes one mistake for every 10,000 bases inserted. Adding on to this, a mutation only occurs once for every 1 billion base pairs that are replicated. In short, the chance of a mistake occurring during DNA replication is extremely slim, but numerous effects will undergo if it happens. And there you have it. That's the process of DNA replication. Overall, DNA replication is a somewhat complicated process, and if rushed, can result in various consequences. Scientists are still discovering new information regarding the process, and that's only the beginning. Thanks for watching.